Hello and welcome to another Mr. Carter Science Special. Thank you for supporting this channel and watching these videos. Don't forget to like and subscribe. In this video we're going to be looking at the immune system, chemical barriers that your body has that help to prevent infections and disease. By the end of this video you'll be able to explain how your white blood cells protect you from disease. So pathogens are disease-causing microorganisms, and pathogens will make you ill by reproducing rapidly, as you can see in the video below, damaging your tissues and producing toxins. The immune system is made up of different white blood cells that fight and destroy pathogens that have invaded and entered your body tissues and the blood. But how do these white blood cells recognise the pathogens as being foreign cells? Well, all cells and viruses have small protein molecules on their outer surface, and these proteins are called antigens. You can see here that the white blood cell has some square antigens, and our pathogenic bacteria cell has some triangular shaped antigens. White blood cells can recognise these foreign antigens because of the different shape, and therefore instigate and start the immune response. There are two different types of white blood cells. There are the phagocytes and there are lymphocytes. You can tell the difference between these based on the shape of their nucleuses in the cells. Phagocytes have this strange lobe-shaped nucleus, as you can see in the cell here, and lymphocytes have a very large circular shaped nucleus, as you can see in the cell here. How do phagocytes destroy and fight pathogens? Well, the phagocyte cell, when it recognises a foreign antigen, will move forward. Its cell membrane will begin to surround the pathogen, and eventually the phagocyte will ingest the pathogenic bacteria, release a number of enzymes which will digest the bacteria and destroy it. This is known as phagocytosis. For your exams, you merely have to say that phagocytes will surround and ingest bacteria in a process called phagocytosis. Lymphocyte white blood cells have a different method to fight pathogenic bacteria and viruses. Lymphocytes produce antitoxins, which will neutralize any toxins produced by the pathogen. Lymphocytes produce antibodies, which will attack and destroy the pathogen, because the antibodies can bind to the antigen on the pathogen's surface, and this reaction will either make phagocytosis easier by clumping large numbers of phagocytes together, clumping large numbers of pathogens together, or sometimes the antibody itself is enough to destroy the pathogenic bacteria and destroy that cell, thus keeping you safe from that pathogen. So as you can see on the diagram to the left, when a pathogen enters the blood, the lymphocyte with the correct complementary antibodies will begin to divide by mitosis. When you are born with a large range of lymphocytes that have the specific complementary antibodies to almost any antigen, but you have very low numbers of these cells, just one cell for each antigen. So when one of those cells matches the complementary antibody to the foreign antigen, those lymphocytes begin to divide by mitosis, and this produces a large amount of identical lymphocytes. Now, some of these lymphocytes will turn into memory lymphocyte cells. However, most of these lymphocytes will be active, producing the correct antibodies and destroying the pathogen. These two different types of lymphocyte cells are very important when we consider how you become immune to a disease. The process I've just described is known as the primary immune response. During the primary immune response, a fairly limited low number of antibodies are produced that have had the effect of destroying the pathogen. However, this primary immune response can take quite a long time, up to 10 days from when the pathogen first enters your body, for the pathogen to have reproduced rapidly enough to produce enough pathogenic cells that one of them comes across the correct lymphocyte with the complementary antibodies to this foreign antigen. It can then take up to another 10 days for that lymphocyte to be able to divide by mitosis and produce enough antibodies that it can destroy the pathogenic infection. When all the pathogens have been killed, some of the lymphocytes with antibodies that match the pathogen's antigen will remain in your blood, and these are called memory cells or memory lymphocytes. If the same pathogen enters your body again, the memory lymphocytes will produce a much faster and much bigger secondary immune response. You will produce more antibodies in a shorter amount of time. This will stop you from becoming ill. You are now immune to the pathogen. This is known as the secondary immune response. So let's just compare those two responses again. The primary immune response. When a pathogen infects a person, lymphocytes recognize the foreign antigen on the pathogen and begin to multiply. Some of the lymphocytes produce antibodies which help to destroy the bacteria, and some of the lymphocytes become memory cells which remain in the body for years. It takes time for these cells to multiply and produce antibodies, and this is a period known as your sickness. This is when you are sick. The secondary immune response. If the pathogen enters your body for a second time, the memory cells recognize the foreign antigen and reproduce rapidly, that's a key mark in your exam, and produce a lot more antibodies, and this will destroy the pathogen before they can harm the body. This secondary response is known as immunity. Thank you very much for watching this short video on chemical barriers and immunity. Let me know in the comments below what you've learnt. Don't forget to like and subscribe, and thank you for your time.